Good afternoon, everybody, and welcome to our Connect Science series. First, just a few housekeeping notes I'd like to start with. Please remember to submit your questions through the Q&A and not the chat session. And at the end of the talk, I'll be able to pull up those questions and ask them on your behalf. I also want to remind you to please check the website that you see on the screen here, and you can find the upcoming talks as well as the recordings of past talks. So with that, it is my distinct honor and pleasure to introduce not only a great colleague, but a dear friend of mine, Dr. Sheila Stewart. Sheila Stewart is the newly minted Gertie T. Corey Chair and Professor of Cell Biology at Washington University in St. Louis. She's also the Associate Director of the Siteman Cancer Center there. Dr. Stewart did her training, her postdoctoral training in Bob Weinberg's lab at the Whitehead Institute. And there she really made some inroads and important insights into tumor intrinsic properties that account for cancer progression, namely looking at how bypass of senescence can lead to tumorigenesis. And then when she moved to Washington University and took her faculty position, she started looking outward at the tumor microenvironment. And she's very well known now in the tumor microenvironment field for her work to understand how senescence and aging, programs associated with aging, affect cancer progression. So with that, I would pass the screen to Dr. Stewart. And again, it's our distinct honor, and thank you so much for accepting the invitation today.
Thank you, Sandy. And I really wanted to give a shout out to both Nellie and David for starting this virtual seminar series. It's been really great. And I was really honored when Sandy invited me to talk, and also thank Claudia for keeping us all organized. And most importantly, all of you who've Zoomed in. I think we're all suffering Zoom fatigue, but it's really been a great medium to try to keep the scientific dialogue going forward. So thank you very much. And I look forward to your questions. So what I'd like to do today is really describe data from our lab that is focused on how age-related changes in the tumor microenvironment contribute to tumorigenesis. And our interest in this is really derived from this graph here. Age remains the single largest risk factor for the development of cancer. And of course, when one sees this type of a graph, you need to ask the question, what is it about the aging process or aging itself that could contribute to such a rapid increase in tumorigenesis in the 5th to 6th decade of life? I think the answer is quite complicated. It's a complicated disease. And I think most of the people on this call today would admit that the vast majority of human cancers are really the accumulation of spontaneous mutations. And we know that in the face of robust DNA repair mechanisms, which all of our cells have, it's just going to take time, not just for those mutations to accumulate as a function of age, as we know they do, but for the right combination of mutations to occur in an incipient tumor cell. So the question really is that it? Is that the driver? And as a card-carrying tumor microenvironment person, I probably would argue no. There are also changes that need to be happening within the tumor microenvironment that contribute to this transformation process. And our interest is really focused on the potential role of senescence or senescent cells. And it's important to note that as with mutations, senescent cells do accumulate within every tissue that has been looked at within the human body as a function of age. And our interest in really looking at the cell derives from the fact that they are able to recapitulate nearly every phenotype that has been ascribed to a cancer-associated fibroblast or a CAF. They can contribute to tumor proliferation, as well as driving increases in migration, invasion. They can contribute to angiogenesis, and they're also able to modulate the immune response and create immunosuppressive environments that we think, and many others do, are able to contribute to that transformation and progression process. So what's the data that would argue in a spontaneous setting that these senescent cells really are important for transformation? I think the best data derives from a paper that was published by Darren Baker when he was in Jan van Dersen's lab a few years ago. And I'm not going to go deep into the genetics of the mouse that they created, but suffice to say this animal has a suicide gene that's inducible. And through a number of different experiments I'm not going to show, they've been able to demonstrate that the induction of this particular transgene is specific to senescent cells. And so when these animals are placed on a dimerization drug, referred to as AP here, what you can see is not only do the mice look better, if you look over here on the right, but if you look at these two graphs, it doesn't matter if you're a male or a female mouse. It doesn't matter if you're on a mixed background or on a straight B6 background. By eliminating senescent cells, you significantly delay the onset of tumorigenesis. And so I think this itself argues strongly that in a spontaneous setting, senescent cells are important for transformation. Now, where these senescent cells are and how they contribute, I suspect that answer is also quite complicated, but I think the question you really have to ask yourself is, how could they be contributing? And I think one of the answers, certainly not the only answer, derives from the fact that senescent cells, in essence, make everything yummy. You can imagine if you're a tumor wannabe. They express large numbers of mitogens. They express factors that can modulate directly the ECM. And they also express a large cadre of inflammatory cytokines and chemokines. And I'll remind you that the second largest risk factor for the development of cancer after the bad luck of getting old is chronic inflammation. So we wanted to actually ask the question, do these cells not only contribute to spontaneous initial tumor formation, but could they also contribute to progression? And so one of the questions we wanted to ask was it possible that the accumulation of senescent cells within the pre-metastatic niche, and here, because of our interest in breast cancer, we've focused on the bone, which is the largest or the most common site of metastasis for breast cancer. And certainly within humans' bones, that's what's shown up here on the right, we can show evidence of senescent cells. Is it possible that the accumulation of these cells over time actually fertilize the soil, the pre-metastatic niche, if you will, to allow dissemination and eventual outgrowth of metastatic lesions? Now, I think this was a few years ago when we published this, but it was kind of important to ask the question because the majority of the data that looks at the pre-metastatic niche have come from beautiful studies from David Leiden and many others that have shown that actually primary tumors secrete factors that then mobilize bone marrow derived cells and can, if you will, fertilize the pre-metastatic soil. And as I just said, those were beautiful studies, but our question was asking, is it possible that the accumulation of senescent cells within the pre-metastatic setting, regardless of a primary tumor, would it be sufficient to drive increases in metastasis? And I'm just going to show you one piece of data because this was published a few years ago. But to ask that question, we created an animal in which we could specifically induce senescence through the overexpression of the cell cycle inhibitor P27 in the absence of a tumor, and then ask whether or not we increase metastasis. In any case, using this animal in which we could specifically induce senescence within the stromal compartment, again, in the absence of a primary tumor, what we were able to show is in this fast mouse where we had increased expression of senescence, we had significantly more metastasis. And basically what we were able to show a few years ago was that we were indeed inducing senescence on the bone lining cells, in this case, osteoblasts. These are the cells that make your bone, if you will. And when we induced senescence, they expressed, again, that large cadre of senescence-associated secretory phenotype proteins, or SASP. And those SASP, while being very complicated expression profile, did express factors that led to an increase in localized osteoclastogenesis. So we had increases in osteoclasts directly surrounding our senescent osteoblasts. And I'll remind you that the osteoclasts eat the bone or resorb the bone. And what happened in this situation is we could show tumor cells specifically seeded to these sites of increased osteoclastogenesis, and this contributed to the increase in the metastasis that we saw. Now, what was interesting is, as I just mentioned, there are many factors being expressed, but one factor that was highly expressed by our senescent osteoblasts was IL-6. And IL-6 is sufficient when there's rank ligand around to increase osteoclasts. And so we could show if we eliminated IL-6 through neutralizing antibodies, we removed that increase in osteoclasts, and we also reduced that increase in metastasis that we had seen in the presence of the senescent osteoblasts. Now, IL-6 was an interesting factor because it really got us thinking about what was happening not only in that metastatic setting, but also in the primary setting in human patients. And so the reason we had been interested in that is because several years earlier, we had shown when we were trying to understand how these SAS factors were regulated, we had shown that these SAS factors were regulated partially by the P38 MAP kinase pathway. And what what P38 is able to do is activate the stabilization of many of these SASP cytokines. But what was interesting is that we saw significant overlap between SASP factors that were found specifically in the stromal compartment of breast cancer patients. And as I just mentioned, many of those regulated by P38. Here I'm just showing IL-6, although I could show you other factors. And what we could show is IL-6, while being expressed within the stroma or the tumor compartment, we also saw significant expression both in the primary setting and in the bone lesions of patients, regardless of subtype. And so it raised the possibility that we could target P38 and use it to limit metastatic growth. Indeed, we had shown, at least in the primary setting years ago, that using either shRNAs or CRISPR to knock down or knock out P38 in our senescent fibroblasts, we completely eliminated their ability to increase tumor growth in the primary setting. We also had a specific drug that we could use to limit this, and we showed that that drug limited the pro-tumorigenic capacity of CAFs as well. And so we wanted to go back to that model that I'd already introduced and ask, in a metastatic setting, could we target P38 to limit the SASP and factors expressed in CAF cells to limit metastatic outgrowth? And so I'm only going to show you data from one cell line, but we've done this now on many different breast cancer lines. And basically the way the experiments are always carried out is we deliver the cells to our animals, and then we wait a day or two to randomize the animals onto treatment. And what we're trying to do here is to recapitulate that early metastatic situation where patients might show to the clinic where they have early dissemination of tumor cells. In this case, we randomized our animals into what I'm going to call for today standard of care, or PTX, paclitaxel, or the P38 as a single agent. Again, our interest was specifically in the bone, but we got an interesting surprise that the effects we saw were both within the bone, which I show on the right, as well as in the visceral organs. So in the interest of time, I'll just describe the data here on the right here with the bones. What we're seeing here on the y-axis is basically tumor growth. These animals their tumor cells express luciferase. And what you can see is that P38 as a single agent was as effective as paclitaxel at limiting tumor growth within the bone and, as I mentioned already, within the visceral organs. So this was pretty exciting. It suggested that this inhibitor was potently able to limit tumor growth. But again, this is a drug. The entire animal's body is flooded with the drug. And I started this talk out suggesting that the target of these P38 inhibitors would in fact be the stromal compartment. So to address that directly, we did two different experiments. On the right, an in vitro experiment in which we show, again, this is now looking at tumor growth within the y-axis, that paclitaxel, as would be expected, limits the growth of these tumor cells. However, when we use the P38 inhibitor that we were working with, these tumor cells don't even blink, they grow fine. I think more convincingly, though, is here, which we've shown here on the right, where we went in with shRNAs that limit P38 expression within our tumor cells specifically, and now introduce those or the RFP listed as control cells here on the right. Again, intracardiac inject to deliver these cells to throughout the body systemically, both to the bone and the visceral organs. And what you can see is that limiting P38 within the tumor cells themselves has no effect on their growth. They grow just fine. And I think these data together, in vitro and in vivo, suggest that the primary target of our drug is in fact the stromal compartment. We were pretty excited about this, but one of the things that we were a bit concerned about is there's been some beautiful data by Julio Guizzo and others that have suggested that the P38 pathway plays an important role in enforcing dormancy, likely through the ATF protein. And what we wanted to try to do is actually get more proximal to the phenotype we were chasing, which we assumed was the stabilization of the SASP messenger RNAs, which I guess I could also call CAF messenger RNAs in this case. And luckily for us, the company that we were working with in St. Louis had developed a novel inhibitor of MK2. And so we wanted to go back and ask, could we, instead of targeting P38, focus our attention on the MK2 kinase? So to do that, first repeat the experiment that we'd done with the P38 approach, which again is to knock down MK2 within our tumor cells specifically, introduce these into animals in the absence of drug, and again show, as with knockdown of P38, when we limit MK2 expression in tumor cells, they grow just fine. Basically exactly the same as control cells. So the tumor cells themselves don't care whether or not they have P38 or, in this case, MK2 expression. However, when we took wild type cells and injected them to animals and randomized those animals now onto an MK2 inhibitor, what we saw was similar to what we had seen with the P38 inhibitor in that we could significantly limit tumor growth in this setting. So this really set us up to try to figure out how it was these drugs were limiting the growth of tumor cells within the metastatic setting. Now, one thing that was interesting is while we were able to significantly limit tumor growth, our ability to increase survival was only modest in this situation. Now, we could argue this is an incredibly aggressive tumor line, and it is. Most animals need to be sacrificed before 13 days of tumor growth, but we wanted to start focusing on whether or not we could leverage these P38 or MK2 inhibitors and the new explosion of immunotherapy, which as to date hasn't been that effective in the breast setting. And one of the reasons we wanted to focus on that is the P38 pathway has a very long history in modulation of the immune response. In fact, it's known that it can modulate dendritic cells' ability to stimulate T cells. Classically, it was originally described in its ability to control cytokine release from LPS-stimulated macrophages, and it also has effects on Treg differentiation. And so just to start to get a flavor for whether or not we were in fact changing the immune response with these drugs, we first went in and looked at what was happening in the adaptive system, or specifically at T cells. And we were able to show, perhaps not surprising, that whether the animals were on a P38 inhibitor or an MK2 inhibitor, and looking both at CD4s as well as CD8s and a variety of different measures of activation, we could show that in these animals, particularly looking at the bone in this case, we saw activation of both CD4 and CD8 T cells. And so before trying to go down this immunotherapy approach, we wanted to ask, are these T cells important for the control of tumor growth in animals treated with either of these inhibitors? And so here's where we got our first surprise. Basically, when we look, and I think a picture is worth 1,000 words, so maybe looking on the right here, but the data is graphed on the left. What you can see is when our animals are put on that MK2 or P38 inhibitor, as I've already shown you a few times, we have a very good limit of tumor growth at the metastatic setting. However, when we deplete CD4 and CD8 T cells, we completely lose the ability to limit tumor growth in the metastatic setting in animals treated specifically with P38. However, this is distinct from the MK2 inhibitor. So even though I've drawn this as P38 limits MK2, limits tumor growth, we got a surprise. There's clearly a difference between the MK2 inhibition, the mechanism of inhibition with MK2 versus P38. So we wanted to go back and dig a little bit further into this and look at CD4s independent of CD8s, expecting CD8s, of course, to be the important pointy end of the stick, as David DiNardo likes to say. And so we went back and again had another surprise. When we singly depleted CD8 cells from animals treated with our P38 inhibitor, where normally we see inhibition of tumor growth, we still inhibited tumor growth. So even though T cells were important for the activity of P38, it wasn't the CD8 cells. Instead, it was the CD4 cells. So when we deplete CD4 cells from our animals and then treat them with a P38 inhibitor, as you can see by the pictures, we have no impact on tumor growth. We completely lose the tumor inhibition ability, of the P38 inhibitor. Now we've gone on beyond neutralizing antibodies and basically show the same with a TCR and alpha knockout animal, as well as an MHC class 2 knockout animal. And so we feel pretty confident that the CD4 cells are very, very important for the P38 inhibition, but we don't know how that's working. Okay, we know CD8 cells aren't in play based on our depletion experiments, but really what's between the P38 inhibition and CD4s and how they limit metastatic growth, we really don't know. And why is it different with MK2? Again, we don't know. But to try to start to dig into this and get at some targets, we decided to start looking first of all at what's happening inside our bone lesions. And the first thing that we did is just to do some basic IHC stains. And what I can show you here is looking at one of our bone lesions from animals treated either with vehicle or the P38 inhibitor. We're not changing infiltration of CD4 cells. So it seems like there's the same number of CD4 cells in an animal treated with vehicle or with P38. However, one of the things that we are seeing when we look in these animals is we do see a significant increase in MHC class 2, again, specifically in animals that have been treated with the P38 inhibitor. So where's the MHC class II coming from? Well, the most likely candidates, of course, are going to be the professional antigen presenters, either dendritic cells or macrophages, although other cells could be responsible. So we decided to try to dig into this and ask, are we seeing an increase in either of these cells, and could they be important for the anti-tumor effects that we were seeing with CD4? So the first thing that we wanted to look at were dendritic cells. There was a paper published a few years ago from Jing Yi's group that had shown that P38 inhibition actually leads to an increase in dendritic cell differentiation. And so I'm not going to, in the interest of time, I'm not going to go through all the data, but what we were able to show was that when we treat dendritic cells before differentiation with a P38 inhibitor, we could show that we do see an ability of these cells in vitro to activate CD4 T cells. In addition, using the ZIA screen antigen tracking approach, we are also able to show that we see increased antigen trafficking to the draining lymph nodes, again, in animals that have metastatic lesions. These things are certainly in play, but are they important? And how is the activation of CD4 T cells, again, important for controlling tumor growth? It's actually quite difficult to start asking these questions. We can continue to do a lot of IHC staining, but how do we actually figure out what's going on inside a metastatic lesion, particularly in the bone, which is our focus here? And so to do this, we decided to turn to an elegant system that was developed by Alara Melichai a couple years ago, actually, and this was in a Nature paper she published. And what she was able to do was to create a system in which she has tumor cells that are expressing a soluble M-cherry. And through a number of experiments she's done, and some that we've done within our own lab, she's shown that this is very limited in the diffusibility of this particular fluorophore with the tag. But what it allows you to do is to label neighboring cells. And in fact, what it would allow us to do was to take a complex organ like the bone and remove the cells inside and isolate stromal cells that are physically inside the metastatic lesion versus those that might be outside it. And so we took this approach using the exact same paradigm that we've used to do our tumor studies, that is, introduce cells into the animals via intracardiac injection, randomize them onto their various treatments, take them through endpoint, and then use bone digestion and flow cytometry to isolate cells that are either positive for M-cherry or negative for M-cherry, isolating tumor cells out away from this. And then ask the question, if we take these cells out, can we perform CyTOF, which we've done? Can we perform RNA-seq and begin to identify them and ask how they're changing in the context of drug treatment? Just to show you a couple controls from this experiment. So these are basically flow cytometry. And what we've done now, again, is take those bones, isolate cells in mass, and then in the case of CD4 cells, which I'm showing you over here on the left, what I can show you is that inside the tumor lesion itself, whether we're looking at animals treated with vehicle or with the P38 inhibitor, we don't see any more or less T cells. It's basically the same. And that's actually what I had shown you with our IHC. So that was pretty exciting. The other thing that we've been able to do is, again, looking now at CD4 T cells that do not contain cherry, so they are outside and physically away from the metastatic lesion, versus T cells, CD4 cells here, that are physically inside the lesion, or at least close enough to it to pick up the cherry. What we can show is we do see CD4 activation, and it's increased in animals that have been treated with the P38 inhibitor. So this data was actually really reassuring for us as we moved forward towards the RNA-seq, and that we were recapitulating things we had seen by our IHC analysis. So I'd like to be able to tell you today the mechanism and what's happening. Unfortunately, we're not there. These data are fairly new, but just to show you that we've got the system up and running and have a ton of data to try to wade through. This is our first RNA-seq, looking specifically now at CD45 positive cells that have been isolated from either animals treated on vehicle in this case, or with our P38 inhibitor. We also have those treated with the MK2 inhibitor that I'm not showing. But importantly, looking again at cells that are physically inside the lesion, this would be cherry positive, versus cells that were outside the lesion, this would be cherry negative. And what's interesting is one of the populations that have popped, i.e. we see a significant increase both in vehicle treated as well as M-cherry, is this kind of bluish population, and these are in fact macrophages. And also what makes us quite happy about these data is when we look specifically at these populations, we see that when treated with a p38 inhibitor, we see significant increases in MHC class 2. Again, what we had already shown with the IHC approach. And in addition, looking at a variety of different cell types, including CD45 negative cells that I'm not showing you, we see significant increases in signatures of gamma interferon, which again, given the increase in the MHC class 2, given the CD4s that are involved, all of this makes sense. Now, again, I've already told you, unfortunately, I can't tell you how this is working, but this has really set the stage now for us to begin to try to dig into the mechanisms responsible for p38's ability to limit tumor growth, again, within the metastatic setting. Now, I showed this graph to you before. I told you that the p38 and MK2 inhibitor greatly limits tumor growth. I've shown you that data and has a modest ability to extend lifespan, or lifespan. I was speaking like an 18 person, but extend tumor-free survival in these animals. But the other thing that was interesting about these animals is when you just looked at them, they're actually much more mobile than animals treated with paclitaxel, okay, which basically has about the same expansion in survival compared to vehicle-treated mice. Now, what I haven't told you is with this particular approach, when we introduce cells into the intercardiac space and deliver them to the bone, usually we have to sacrifice the animals because they basically break their legs, okay? And that's because they have such terrible burdens within their bones. So our humane endpoint tends to be when they lose mobility. And what we noticed is, again, as I just said, the p38 and MK2 treated animals, unlike the paclitaxel treated animals, were much more mobile running around. And so this got us thinking about what's actually happening in our animals, but more importantly, what's happening in patients. So bone, I've said it 3 or 4 times now, is the most common site of metastasis for breast cancer. And unfortunately, once tumor cells have made it to the bone, this usually is considered an incurable disease. Many of the therapies are palatable in nature. But one of the reasons that this is so terrible for patients is it causes a lot of pain. Often you'll see spinal cord compression problems in these patients, but you'll also see a high rate of pathologic fracture in these patients. And the reason for this really is the fact that tumor cells are acutely able to induce bone loss. So it's known as the vicious cycle. Tumor cells arrive in the bone. They release factors that then drive increases in osteoclastogenesis, which it basically opens up real estate, if you will, but more importantly, releases large amounts of latent growth factors that are locked in the bone parenchyma, which tumor cells are able to use to increase their growth. And so again, this is usually thought of as the vicious cycle and basically a feed-forward loop, if you will. Now, this has been dealt with very well in the clinic by the introduction of the bisphosphonates as well as denosumab. And this is actually, these 2 drugs together or approaches have reduced these pathologic problems in the bone by over 1/3 in patients across multiple different types of tumors that do actually metastasize to the bone. The way in which this works is that at least the bisphosphonates is what I'll talk about today. They covalently link or attach themselves to the bone matrix or the mineralized bone. And as the osteoclasts are eating their way through the bone, resorbing the bone, this kills them. It basically allows them to activate it. Unfortunately, while this protects the bone or the patient from bone loss, other than in the postmenopausal setting, which is still a bit confusing as to why that would be so different, this doesn't do anything to slow down tumor growth in patients. So we got interested in thinking about this, not just because our animals are running around more, but because we knew, based on publications from many other groups, that p38 and MK2 actually play a very important role, not only in osteoclast differentiation, but also in their ability to resorb or to eat bone. In data I'm not going to have time to go through, we've gone back and shown that in an in vitro setting, both of our inhibitors are quite capable of both inhibiting osteoclast differentiation and also inhibiting resorption of actual bone by osteoclasts that are formed. So what's happening in the bones of our patients, if you will, our furry patients? So here I'm showing you data. Underneath, you can see these are reconstructions of the bone where we've taken the femur and you're looking straight through it. I'll remind you, for those who don't think about the bone, the femur, obviously, you have your hard cortex, and then through the middle, you have this webbing, which is the trabecular bone. It really provides flexibility, if you will, to the bone. And so basically what we did was to measure the density of the bone. I'm showing you here on the y-axis trabecular BVTV. Just think of it as density. I'm also showing you what the BVTV is of a non-tumor bearing mouse on this strain background, which is about 0.3. And the reason that I'm showing you these data opposed to data from a vehicle treated animal is I would be forced to show you fake data, and I don't like fake data. But I'm going to go ahead and give you this oval down here near 0. Basically, these animals that are on vehicle or have no treatment, they have such huge losses of bone that they tend to have huge holes in the bone, and we can't measure the contours to actually get an accurate measurement. This is also why they become immobile and their bones break. So we're going to put this little oval here as a reference point, but this is where the animals start out with these red circles here in the non-tumor bearing lane. So if we look now at animals that are bearing tumors, animals that are on paclitaxel or PTX, you can see that they lose significant amounts of bone. However, we are able to control tumor growth to the same extent as what we were able to do both with the P38 and the MK2 inhibitor. However, if we look at the bones of the animals that have been randomized onto either the MK2 inhibitor in these diamonds or the P38 inhibitor in the triangles, you can see that these bones are basically wild type, okay, same as our non-tumor bearing mice. And so we think that this is one of the major reasons these animals remain more mobile throughout the experiment. Now, what's interesting to point out here, though, is when we take animals and instead of putting them on the P38 or the MK2 inhibitor, we put them on zoledronic acid, which is one of these bisphosphonates. As you see in patients, we are in fact able to stabilize the bones of these animals. However, as we see in premenopausal women, we do nothing to slow tumor growth down. So even though we can protect the bones with zoledronic acid, we cannot slow the tumors down. And so we like to think of the P38 and MK2 inhibitors, if you will, as a two for one. On the one hand, these inhibitors are able to directly block the stromal compartment's ability to support tumor growth. Obviously, I need to remake this slide because P38 and MK2 seem to be doing this in quite different ways. P38, as I've just shown you, requires CD4 T cells in ways we're still trying to understand, whereas MK2 inhibition does not. But in addition to that, the P38 and MK2 inhibitors are able to also limit osteoclastogenesis and thereby protect the bones. So the question that this really started to pose for us is, what is it that's driving the extensive bone loss that we actually see in patients, right? I told you a few slides ago that certainly once tumor cells have arrived in the bone, this vicious cycle is part of it. It's pushing increases in osteoclastogenesis, which are then allowed to resorb bone if that remains unchecked with things like bisphosphonates. But this is actually happening in more than just metastatic patients, all right? Bone loss is also seen in patients with primary disease. And the question is, why is that? What's driving this bone loss? Well, part of the answer is estrogen. Estrogen, whether you're a male or a female, plays a very important role in helping to preserve bone integrity or density. And the importance of estrogen is underscored if we look at patients, well, I shouldn't say patients when I'm talking about menopause, but if we look at women that are either in late or early menopause, you can see that they lose upwards of 2% of their bone density per year if left unchecked. And if you look at breast cancer patients that are on aromatase inhibitors, remember aromatase is an enzyme in the biosynthetic pathway for the creation of estrogen. This is used to deprive estrogen-dependent primary tumors, if you will, from estrogen. These patients also lose bone a little bit more than you see in patients with in menopause, and this is because this is going to limit all estrogen production. So estrogen plays a very important role in bone density. And this is not just in humans, this is true in mice as well. If you take animals and you subject them to OVX, remove their ovaries, give them hysterectomy, if you will. And again, now here we're looking at bone density. What you can see is that these mice, these furry patients, lose bone as you would see in a woman who's entered menopause. Okay, so this is something we can model in the animals as well. And so how does estrogen do this? The answer is complex. Estrogen plays important roles in osteocytes. These are the nerves of the cells, if you will, that are important for detecting mechanical signals that move through the bone and talking to other cells like osteoblasts and osteoclasts. But estrogen also has direct roles on osteoblasts, Those are the cells that make your bone. I've already talked about those. Osteoclasts, the cells that resorb or eat your bone. And interesting, also affects T cell biology that again has effects in osteoclasts. And so the idea out there has been that if you look at patients that are being treated with, for example, chemotherapy, we know that chemotherapy can induce a premature menopause within patients. This, of course, is going to lead to an estrogen deficiency. And given what I've shown you with the OVX, given the importance of estrogen in the bone, is going to contribute to bone loss. So that's really kind of where things have stood for a long time. But the problem with this is really when you go back and you look at patients. So the first few bars here, I just showed you a few slides ago, trying to argue that estrogen is in fact important in patients. But we were always befuddled, if you will, by the bone loss you see in the blue box here, right? If estrogen is the sole driver, I should say loss of estrogen is the sole driver of bone loss in patients that are being treated with chemotherapy, why are they losing so much more bone than a patient that is simply deprived of estrogen through aromatase therapy? And so we wanted to start thinking about whether we could model this in our animals, and we can. So again, I'm showing you bone density, BVTV of animals, and again, the reconstructions are here to the right. These are data I just showed you. If you take an animal and you treat it, or you remove its ovaries, you lose a lot of bone, as seen here. If you take animals and instead treat them with chemotherapy, in this case, we're using doxorubicin, but you could use others. We've done similar experiments with paclitaxel. As with patients, they lose a lot more bone. So chemotherapy drives more bone loss than estrogen deprivation alone. So where's this extra bone loss coming from? Well, I often like to tell my students, if it walks like a duck and it quacks like a duck, sometimes it's a duck. And what I mean by that is chemotherapy is a potent inducer of senescence. When one uses chemotherapy, we see induction of senescence, as I'm showing here. This happens to be osteoblasts that have been treated with chemotherapy. And we've shown when we worked with our fast mouse that I had talked about at the very beginning, that when we induce senescence with chemotherapy or overexpression of P27 in osteoblasts, we see significant increases in a large number of SAS factors. Here I'm just showing you a few of them. Again, this is specifically looking at osteoblasts. In addition, osteoblasts lose the ability to mineralize bone. And so there really are cells in these other empty wells, but this red color is mineralization through osteoblasts, and this lower one is a panel, and I'm just showing, this is all published, I'm just showing for example, but basically we completely lose mineralization capacity when osteoblasts enter senescence. In addition, when these cells enter senescence and express these SAS factors, IL-6 being one of the important ones, we actually see significant increases in osteoclastogenesis. And so these large kind of goobers, if you will, pink messes are syncytia, which are osteoclasts. And these are the size of osteoclasts and numbers we see in normal vehicle-treated osteoblasts. But when we have senescent osteoblasts, we see significant increases in osteoclastogenesis. And so this raised the possibility that in addition to estrogen loss, perhaps treatment with chemotherapy is leading to senescence within the bones. And this is in fact contributing to the bone loss that we see in at least in our animals. So to try to address this and ask if this was in fact true, what we did was to take animals, and here I'm just showing you the doxorubicin treated animals, although I think I mentioned we've done this with paclitaxel as well. And the first thing that we've done is, this is a histologic section doing a stain for the senescence-associated beta-galactosidase, which shows up as a blue stain. And just to kind of orientate you, what you're seeing here is these, I'd like to call them white rivers, if you will. These are that trabecular bone, that mesh that I was talking about that runs through the bones. And then obviously all of these smaller pieces or dots here are bone marrow-derived cells, immune cells, if you will. And what you can see is we see a significant increase, which is quantitated underneath, in SA beta gal positive cells in animals that have been treated with doxorubicin versus animals that have been treated with vehicle. And if we take these cells, or if we isolate cells from the bone of these animals and run qPCR, we can also see a number of factors that are consistent with the induction of senescence. For example, P16 is increased in animals treated with doxorubicin. This is a cell cycle inhibitor that we all often see associated with senescence. We see reductions in HMGBM1, again, consistent with senescence, and increases in a large number of the SAS factors. I'm just showing you a few. And importantly, IL-6 is up, which I think I mentioned at the beginning of the talk, is sufficient when there's rank ligand around to increase or significantly increase osteoclast differentiation. So we could see induction of senescence by SA beta gal and as evidenced by SAS expression within the bones of animals treated with chemotherapy. But does it matter? So we were lucky enough to collaborate with Darren Baker, who created this ink attack mouse. I described it very briefly at the very beginning of the talk, but just to reiterate, the way that this mouse works is there is a P16 promoter that's driving an inducible suicide gene. And so when AP is delivered to these animals, it's allowed to activate a caspase that is expressed, leading to the death of senescent cells. And there's been a rich history of papers published on validating this approach. So I'm not going to show you any of that data. You can look it up and I'm happy to discuss it. But importantly, what we were able to do is take our animals, treat them with chemotherapy, and then eliminate senescent cells and ask what's actually happening. So the first thing that we noted is that when animals are treated with AP, so animals are receiving doxorubicin, we treat them with AP to initiate this caspase and kill senescent cells. You can see we lose expression of P16 and expression of IL-6 as well as other SAS factors and increase HMGB1. So we think that this supports the idea or argues that in fact we are inducing senescence. I showed you that in the last slide, and we're able to eliminate at least the senescence effector arm, if you will, parts of the SAS. So what does it do for bone? So to do that, we basically repeated the experiments we've done before, treating our animals with doxorubicin, now in the presence or absence of the dimerizer, i.e. either killing senescent cells or not. And what we were able to show was that the significant bone loss that we see upon treatment with chemotherapy, here listed with doxorubicin, actually we're able to rescue a fair amount of that when we treat the animals with AP. In other words, when we eliminate the senescent cells. Supporting our argument that senescence does play an important role in at least some of the bone loss we see in with the treatment of chemotherapy. Now, I'm not going to go through and show you the data in the interest of time as we're running short here, but we've gone on to show that when we eliminate senescent cells, so looking here at these upside down gray triangles, this leads to a significant reduction in trap activity. In other words, we see a significant reduction in osteoclasts and their ability to resorb bone. And in addition, we see a significant increase in bone formation. In other words, the osteoblasts are mineralizing much more bone when senescent cells are lost. Now, what we don't know yet is really mechanistically, is it we've kind of changed the balance between bone resorption and bone formation, or have we changed the timing of when bone is forming? And I can discuss that if anyone's interested at the end of the talk. So the last thing that we wanted to then ask is, given what we knew about the P38 and MK2 kinases and their ability to contribute to SASP expression through the stabilization of SAS messenger RNAs, could we go back and use these inhibitors to potentially stabilize the bone of animals that are being treated with chemotherapy? And so just to show you at least that the target is on, if we look in the bones of animals that have been treated with doxorubicin, shown here, this IHC up in the upper right, and quantitated underneath below, what you can see is that we see significant activation of the pathway when animals are treated with chemotherapy. Probably not so surprising, but at least our target is on within the bone itself. And so if we take animals now that have been treated with doxorubicin, in which we know the P38 MK kinase has been activated, when we look at what happens to the bones, what we can see is that, again, the bone loss that we've seen with treatment with doxorubicin, shown here in this, these are squares, we're actually able, both in the presence of P38 inhibitor and in the presence of the MK2 inhibitor, to rescue that bone loss. And I think this is quite exciting because if we think about patients that are being treated with chemotherapy, particularly thinking about patients that are in the primary setting, typically these patients don't receive, for example, bisphosphonates or denosumab to protect their bones, despite the fact that they lose their bones. And some of it might be driven by the fact that bisphosphonates, I like to say, are kind of like getting married, right? If you are given bisphosphonates, it's basically in your bones till your osteoclasts resorb through it. On an average human, it's thought to be about 7 years before you turn that over. And of course, these drugs are not without their risks. Osteonecrosis of the jaw, while rare, is a terrible side effect of the bisphosphonates. And one potential advantage of the P38 or MK2 approach is you're taking it when you're taking it, and you're not when you're not. So one of the things that we're quite excited about is we recently received a DoD Level 3 award to actually put the MK2 inhibitor in the metastatic setting in a clinical trial to ask whether we could limit tumor growth, but also protect the bone. And if that trial were successful, it may open the opportunity to think about using these drugs actually in the earlier setting or the primary setting, where we may be able to actually afford these patients some protection from their bone while also limiting tumor progression. So in just the last 2 or 3 slides, we've now tried to start thinking about mechanistically, how is chemotherapy really contributing to bone loss? We know they lose bone when we activate senescence. We know that if we block the SAS, we can protect the bones. And we also know that if we eliminate senescent cells, we increase bone formation and decrease bone resorption. But we don't know that the cells within the bone themselves are responsible for this. I'll remind you that with that ink attack mouse, when we eliminate senescent cells, we're eliminating senescent cells throughout the entire body. And so we've started to do this by first just looking at where the senescence activation might occur. And so this is an experiment that we did to end our paper that we recently published, in which we sorted out cells from the bone, and I'm listing the cell types underneath here, from animals that were either treated with vehicle or treated with doxorubicin. And what you can see is that pretty much the only place we see a significant increase in P16, which again is the driver of our suicide gene, really is in the CD45 negative, CD31 negative population. And so this, of course, includes quite a few different types of cells, but this is where the activation is. And we don't see it, and I think I've taken it out in the interest of time, in the osteocyte population. And this is important because it was published by Kailash Deepa at the Mayo a few years ago, that in fact, bone loss related to aging, so age-related bone loss, is driven by senescence within osteocytes. We don't see that in chemotherapy. Chemotherapy seems to be attacking a different type of cell. So to try to get at this, and I have another teaser for you, and I apologize, we really just got all of these data in the last couple of months. But basically what we've decided to do is try to take that unbiased approach and ask, can we figure out where senescence is happening? And if we can figure out where it's happening, can we start to target those cells and figure out if they're responsible for the bone loss? And so we've basically taken the approach that's detailed here over on the left to isolate bone cells, primarily focusing on cells that are depleted of CD45 and CD31. And I'll remind you that's actually because it's those cells where we see this increase in senescence, as well as getting rid of the erythroid progenitors. Sort those cells and carry out RNA-seq. And I'm just showing you one picture here from a vehicle-treated animal and a large list, and I've already gotten rid of the colors because it's too complicated, but we're actually quite happy to show that we can recapitulate most of the cell types that have been recently described by RNA-seq by David Skadden, as well as Simon Haas, is very interesting paper. So we're able to see most of the cell types that we would expect to find within the bone using this approach. And basically, what I can show you is that if we look at animals treated with vehicle versus chemotherapy versus animals in which we've isolated the same cell population from animals that are 26 months of age, we see significant differences. We are seeing P16 expression in a couple of different populations, primarily what look to be mesenchymal stem cells, as well as 2 fibroblast populations. And we don't see those expressions in the old animals. Now, we don't have osteocytes. We're not actually able to isolate those from the bone and look at that. But at least we now have populations within the bone that we can begin to target once we validate them and ask, are they contributing to the bone loss that we see? And if they are, how are they doing it? With that, I need to conclude with the most important slide, and that is the people that have actually done all of the work. The early FAST work that I mentioned was work from Jinmin Lo in the lab. All of the anti-tumor work and early bone loss work was done by a very talented graduate student who's now moved on to better things, if you will, Bhavna Murali, as well as a postdoc pictured here, Jiangtang Yao. All of the immune work that we've done has been the work of a very talented postdoc, Douglas here, who's trying to hit me with an axe, and helped by Tom as well as Chiho. And I just wanted to also end with one other thing that I have to, because James DeGregory said he's going to watch this tomorrow. But in all seriously, we have launched a new journal that is interested in really bringing together the aging and cancer field. So we encourage you to submit to it if you have anything interesting, so we can get this journal off and launched. And with that, I would be more than happy to take questions.
Excellent. Thank you, Dr. Stewart. Sorry, I had a hard time reconnecting. We have a number of questions, but I have to start with the first one. Why is your lab coming after you?
No, this is just to show you that I absolutely trust my lab.
Good point. Well, let me launch right into the questions because we have a number to get through, and thank you for leaving time. So the first question, would we be seeing similar results in limiting tumor growth for cancers other than breast cancer? That came in very early, so perhaps related to the first part of your talk in limiting breast cancer growth.
Yeah, I mean, we haven't looked, so the short answer is I don't know. Given the propensity of lung and prostate to go to the bone, at least where we've focused in and been able to look at the effects of these drugs on the osteoclasts, which are very important in both those diseases. Maybe, I mean, we'd love to collaborate with someone. You know, at one point you have to focus. So I don't know the answer to that.
Great. Another question. Wonderful talk. Does the systemic regulation of bone loss involve a crosstalk with muscle in these models? Does the muscle secretome change with chemotherapy treatment and impact bone loss? Is there any impact of P38 inhibition or MK2 inhibition on weight loss, muscle loss?
Whoever you are, contact me later. Yeah, it's a $100 million question. We are actually now trying to look both at muscle as well as peripheral neuropathy, which is another major side effect of chemotherapy, and cardiac defects. We are not an expert. So we definitely see, the way we've set this model up, we definitely see muscle loss. And at least our initial work, and I'm just going to say this has been limited to doing one stain of muscle fibers, so please don't hold me to this, is I don't think we're seeing a rescue of the loss with chemotherapy with at least the MK2 inhibitor. We have not looked with P38. But that is something I think is super interesting. This crosstalk, we haven't even begun to delve into it, but we are now trying to do so. So if you're interested, please contact me. We'd love to get an expert on board.
Yeah, great. And that does remind me that, Claudia can send you this list of questions, and you'll know who's asking questions, so that you can reach out if you'd like.
Does that mean they can run, but they can't hide from me?
Possibly. No one can outrun Sheila. I have to just tell you that. Another point and question, great talk. It seems that P38 inhibition and MK2 inhibition act via different pathways. Have you tried combining the two? I think maybe you can restate that. Also, with the CD4 T cell effects of P38 inhibition, have you tried combination with anti-CTLA-4?
Yeah, okay, so two really good questions. We haven't tried combining the two. We have thought about it, and it's more of a logistical issue. So it turns out that with these inhibitors, and people may have seen literature in the past, particularly with P38, it's kind of mixed effects. And when we were using like the SB drugs and other drugs where you have to do injections, we saw really small effects. We've now gone with the company that we work with in St. Louis, Aclaris, and all of our food is compounded into chow. So these animals get a much longer release, and we see these larger effects. And so we thought about it. We just haven't figured out how to actually, I guess we just put it in the same pellets, but it's more of a technical issue. But we do not see the same effects when we do, IP injections once a day. As far as CTLA-4, we have, in fact, you always tell the story where it sounds more interesting, but we actually started with trying to put CTLA-4 or anti-CTLA-4 and PD-1 with the P38 versus MK2 inhibitor. We don't see any synergy with the with P38. We actually see a small bit of synergy with the MK2, and in some ways that's starting to make sense given the CD4 dependence for P38, which we obviously don't understand mechanistically. So short answer, don't see any combinatorial effect with the P38 inhibitor, see a little bit with the MK2 inhibitor.
Interesting. Another question. This kind of goes back to what you've already said earlier about prostate cancer, but it says prostate cancer also frequently metastasizes to bone. Is there a role for androgens in the process you describe?
Yeah, again, we haven't looked. We've kind of purposely focused on estrogen, which is downstream of testosterone, at least, right? But, you know, people always talk about prostate as being an osteoblastic disease and breast being an osteolytic disease. The truth is, a lot of prostates have both in it. So if the osteoclasts are very important in that disease, we would expect a limited effect. Again, because we know we can target the osteoclast. Beyond that, though, we haven't tested it. As I've said twice already, anybody who wants to look at other diseases, we should definitely talk.
Yep. How did you control for the specificity of the P38 inhibitor?
Yeah, so the controls were really to the in vitro experiment I showed you, but I think more convincingly was the shRNA. So when we limit shRNA expression, or limit P38 expression through shRNA, sorry about that, to levels that are similar to what we envision our kinase inhibitors are working, those tumor cells grow fine. If anything, they almost look like they're growing faster. So to us, that argues that the primary target of these drugs need to be X tumor cells, the stromal compartment in some way.
I think maybe, I'm not sure, but I think maybe they were asking about, you know, given the P38 pathway, various pathways through which P38 can act, you know, and targets and effectors of P38, is there specificity in that inhibitor? That may be what they were asking, but I'll elaborate anyway.
Well, there's two ways I could take that, and maybe this person could also email me, but as far as specificity to P38 itself, these drugs, you know, doing kinase assays in a test tube, the specificity for the P38 alpha is extremely high. They see very little effect on 50 other kinases, but who knows what happens to kinase 51, right? And then as far as specificity to other downstream effectors, so the MK2 inhibitor, it does not inhibit the MK2 kinase actually. What it does is it gets in between the pocket between P38 and MK2, and it doesn't fit in the pocket, at least on a, you know, when you look on it on a computer screen, between the other kinases we know of. So from that standpoint, we think that inhibitor is pretty specific for the activation of MK2 by P38.
Cool.
So,
yep, great. Another question, oh, we still have time. You mentioned a mesenchymal bone marrow cell driver of metastasis-associated senescence in the bone. Could you expand on this, and how would this be modified by age? I guess that kind of ties in with the question I was having too about looking at mesenchymal stem cells or MSCs in the bone and wondering, does your fast mouse model hit mesenchymal, you know, bone marrow cells?
Yeah, so do we hit mesenchymal stem cells with the FAST model? Not to our knowledge. That model is driven by a collagen, which shouldn't be on a mesenchymal stem cells, but if single cell RNA-seq is teaching us anything, there are rules and then there's reality. So I don't know. To the person's question, I think if you were asking about MSCs, I mentioned that when we looked at the stromal cells from chemo-treated animals, the places that we see increases in P16 that I think are believable, that we obviously need to validate, are LEPR high cells that are consistent with Skadden's definition of mesenchymal stem cell by RNA-seq, and a couple fibroblasts. And why I call them fibroblasts is because they're FSP high, as well as a few other factors that make a lot of collagen. So they make a lot of P16, we see SAS factors up. So they are our favorite targets right now, but again, that's RNA-seq. There's nothing functional there yet, but we are definitely working on it.
And part of the question too was, what do you know about how these different cell types may be modified by age or change with age? Is that known?
You certainly see in the bone, most of the work has looked at like increases in ROS, mitochondrial dysfunction. But I'd like to point out, I went through this fast and I probably should have left the slide in, but I think what's really interesting is if you read the senescence literature, I either get, you senescence people, you don't have a defined signature. And I'm like, or I get, oh, you senescence people, so cool, you don't have a defined signature. Different cell types are very different. So cells know where they came from and they know that how they got hurt. And what I mean by that is you can take the same fibroblasts and induce senescence with a bunch of different things, and their SAS are very different. You can take the same drug and hit different cells, and their SAS are different, yet they all senesce. So why do I tell you that? Well, if you go and look at this paper that was published by Farr in Nature Medicine that looks at age-related bone loss, they show that the really only signature of senescence that they see is in osteocytes. And when they use that ink attack mouse, they rescue the bone loss that they see in age. When we go back and look at our animals that are treated with chemotherapy, we don't see any changes consistent with senescence in osteocytes. We do see it in the old mice, so we recapitulate what Farr has seen. And instead, our changes are only seen in those few cell types that I've mentioned already, at least in our initial analysis. So while I actually went into this assuming chemo is just a rapid aging event, I think it can be, but I think it's a little more complex and it's going to take time to tease these apart.
Yep. I think we'll have time for one more, and I'll just ask, it's a little conceptual, but maybe that's where you're headed. You kind of touched upon this a little bit. So a lot of your models beautifully recapitulate stage 4 disease when, metastasis is already present, and, finding ways to even preclinically be able to treat those lesions is pretty spectacular. So I wonder if you've done experiments to actually look at pre-treating mice with any of your combination therapies, or monotherapies or combination therapies, and then done the intracardiac injection to see whether you might even prevent metastasis as a way to think of, either in a neoadjuvant setting or follow up, after surgery. Could these therapies perhaps be used earlier? In earlier stage disease?
Yeah, so could we slit on seeding, if you will? Yeah, we focused on purpose at what we're calling the post-seeding. We all have to admit, intracardiac injection is not true metastasis. We're looking post-seeding. And we did that because what we were trying to first do is say, hey, patients are going to show up with a primary lesion. There's now tons of, I shouldn't say tons, but quite a bit of data argue that tumor cells, the horse is out. Tumor cells left long ago. So we know those cells are already out there. So we were really trying to set up a model that looked at that. What happens if you take out the primary and now you're treating? So we haven't done what you've asked. What I think is we should do, but I think the models are just very difficult, is could we take an animal with a primary tumor and then do remove the tumor and look at whether we affect metastasis? We've done a little bit. We haven't had any success with it. Again, whether that's because it's the 1 or 2 tumor lines where this works, and I don't know. So it's a good idea, but we haven't done it. We need to do it. So
great. Well, unfortunately, we're out of time. We do have more questions, but definitely we can forward those to you and stimulate more conversation after the fact. So once again, on behalf of everyone attending and everyone at the Farber, thank you so much for accepting our invitation and presenting spectacular work that really, pleased to see the translation of your work into a clinical trial. So good luck, and thank you again.
Thank you, everybody, and stay healthy.